Hello, everyone. Good evening to everybody. Am I audible? Can you people hear me? Hello, hello, everyone. Good evening. Good evening to you all. All right. So, how are you all doing? If you just remember, let me tell you, my name is Tithi. I had taken a few sessions in the beginning of the course, and after many days, I am coming back. So, how are your classes going on, everyone? Yes, how is it going on, everybody? Very well. Good. Good, Nayan. That is what we expect. And that is why we started this series. Okay. Our intention was to help you guys in every bit. Okay. All right. So I see people joining, but yeah, we will keep them joining and we will start our session. All right. Okay. So today we are going to talk about a topic from molecular genetics. But yes, before going into the topic, let me ask you a very normal thing. Have you people watched the movie X-Men? There is an entire series of movies, old, new, in every time zone you will get a movie from X-Men. Have you people watched any of those movies of X-Men? Yes, Nayan has watched. Nobody else? That's surprising. These X-Men movies are very popular. Wolverine or Magneto. Yes, Jason has watched. What? Do we have very studious people in this group here who don't study, who just study and doesn't want to watch any movie? Okay, Shubhi has watched. Well, I thought all of you will answer that yes, at some point of time we have watched the movie. Okay, anyways, so those of you who have watched X-Men and those of you who have not, so let me tell you what is X-Men. It's a movie about a bunch of people, men and women both, with some extraordinary power. Okay, in the movie they refer to them as mutants. Okay, extraordinary power they have because they have some unique feature in their DNA which makes them mutant, and because of that they have those special characters. Okay, is it not what X-Men about? So today we are going to talk about mutations. All right, Rahul. Okay. So do you people agree? Is it not? X-Men is all about those bunch of guys with some special power because they have some change, some unique feature in their DNA, which makes them, which allows them to have those unique characters or special characters, right? Some people use it for good, some people use it for bad. That is what is X-Men all about. Okay, isn't it? Right. So today we are going to talk about, of course, not X-Men, but yes, we are going to talk about mutations. Okay. So mutations at gene level, at chromosome level, and along with that, we are going to talk about population. Welcome to the session, everyone. So, very first question, what is mutation? So, as I was telling you, mutation can be defined as a change in the DNA sequence of an organism. Okay? Any change in the sequence of an organism can be called as mutation if that change is heritable. Okay? That, that means it is passed on from one generation to another. Okay? Now, it's not that always that is what happens, but yes, mutations can result from errors in DNA replication during cell division, exposure to mutagens, or any viral infection. Okay? Mutagens are mutation causing agents. Okay? So, mutation causing agents are what called as mutagens. Okay? So, mutations can cause because of error in the DNA replication or can be caused because of some exposure to this mutation causing agent. All right. So, that is what causes mutation. Right. Now, still, even though we have seen like in X-Men, everywhere they get mutants, but in real life, do you come across mutants or do you come across mutants that frequently? No, no, we do not come across mutants that frequently, isn't it? Right? But you have to understand here one more thing that mutants are not always like what showed in the movie. Like it's not always they will have a special power. Okay. Even when we talk about the alleles of a gene, which are alternate forms of a gene, they are also derived because of mutation, because of some changes happening in the nucleotide sequence of the organism. Right? So if we see the mutation here, so mutation, so there is a normal gene. Okay, just to show you the effect what mutation can do. That if you have a normal gene, that normal gene is transcribed and translated, and that will form the normal protein product. And that protein product will have a certain normal phenotype. It will produce a certain phenotype which will be normal. But if suppose a mutation happens in the normal gene, then the normal gene has got mutated. Okay? And with that change now, with the transcription and translation of that changed version of the gene, a different product will be formed. Okay? Abnormal here, yes, it is not normal. It is not the one which is usually formed. It is different. Okay? Now, this different protein product will have a different phenotypic expression. All right. So that is what mutation can do. But yes, it is not always that, or it doesn't happen all the time. Like mutation will always alter a phenotype. Okay, and during this session today, you will come across a various different types of mutations. Okay, we'll talk about all of them. Now, before moving ahead, can you people tell me, are mutations good or bad? What do you think? Mutations are good or bad? Can I have some responses? Both. Okay. Yes, absolutely correct, Jason, should be that it is both, right? Mutations can be good as well as bad. Okay. So because of bad mutations will be what that we will some proteins are not at all produced. Because of that, the cell is not even able to survive. Okay? So in that scenarios, the mutation will result into lethality. Okay. So yes, for in those situations it is bad. But if we see the evolution process, okay, so human beings have or Homo sapiens have evolved from the primates, right? So from primates, if we see, all right, so from chimpanzee, from gorilla, so from there, if we talk about Homo sapiens, so we are nothing but a collection of some useful mutations that has resulted in the evolution of human beings, right? So mutations can be good and bad. All right. Now let's talk about the types of mutation. Okay? So types of mutation, the first types of mutation that we are going to talk about will be depending on the size of the DNA lesion. Okay? The size of the DNA that is getting affected. So on the basis of that, mutation can be of two types, gene mutation and chromosomal mutation. Gene mutations will fall when the size of the DNA lesion is so small, okay, that it affects a single gene. Only one gene is affected. Okay, that is what is called as gene mutation. Chromosomal mutation, the size of the DNA lesion will be big enough so that it causes large structural changes in the chromosome, okay, in the chromosome structure or in the chromosome number. Okay. The size of the DNA lesion is relatively large, causing structural alterations of chromosome, and that is what is called as chromosomal mutation. Now, if the gene size of the DNA lesion is small, okay, so it affects a single gene, but then how do we detect it? Definitely, we cannot detect it if we look at it in the if we look at it uh, in the under the microscope, right? By just looking at the structure of the chromosome, we cannot detect the gene mutation. So for that, we have to for detecting it, we have to observe the changes in the phenotype. Everybody, am I audible? Okay, all right. I think Manisha Subhi, you have to check your internet connection. Okay, just have a check on it because other people are saying that the voice is all right. So chromosomal mutations. Okay. So chromosomal mutations, the size of the DNA lesion is big. Now, when it is big, so it is affecting the entire structure of the chromosome. Okay, and that can be detected easily when we look at the chromosome with microscope. Okay, so in that way, chromosomal mutations are easily detectable under microscope, whereas gene mutations you have to check the phenotype of the gene. But the chromosomal mutations are relatively more dangerous. Why? Because its effect will be relatively more because it is not affecting a single gene but multiple genes that are present on the chromosome. Because the size of the DNA lesion is big. Okay? Gene mutations, size is small, so it is affecting a single gene. Okay? But never mind, okay, there are examples where a single mutation in a single gene can also be lethal for the cell. All right. Okay, so before moving ahead, everyone, this is our details. So please download the BioTecNika app for getting more information on the classes, podcasts, webinars, new courses, everything new that is happening in the biotechnology field. You can get an information from the BioTecNika app, which is available on Google Play as well as Apple App Store. And also, you can join our Telegram group, okay, for more information, for more updates on everything that is happening in the biology field. All right, everyone. And as you all know, stay tuned till the end of the lecture. And by giving us your feedback, your valuable feedback, you will receive the lecture notes for the session. All right, moving on. Is my voice better, everybody? Shubhi, Shamalina, Avishka. Is the voice clear now? Is it any better? All right, great. Okay, so moving on. Gene mutations. Gene mutations can be of three types: base substitutions, deletions, and insertions. Okay. Now, what are base substitutions? So, substitution means one is replaced with another. Okay. So, if you have a DNA sequence of this, okay, so if base substitution is happening at this position, that means this G will be replaced with any other base that is there in the DNA. Okay, so DNA has a adenine, thymine, guanine, and cytosine. Okay, so guanine can be replaced with any one of the other base. Then it will be called as a base substitution mutation. Okay. So as you can see here that this G getting replaced with A. Alright. So it will be a base substitution mutation. Deletion will be where a nucleotide is deleted. Okay. Now let me tell you that this deletion mutation we will see in the chromosomal mutation also. Okay, we will see the types of chromosomal mutations and there also the deletion mutation will come. But that time the deletion will be big. Okay, a large segment of the chromosome will be deleted. But in gene mutations, when we are talking about deletion, it will include one or just a few nucleotides. Okay, same with the insertion. So, insertion or deletion type of gene mutation includes insertion or deletion, respectively, of one or few only nucleotides. All right. So, if it is deletion, that means this G got deleted. Okay. If it is insertion, an extra T got inserted between this pair of A and G. Between this pair A and G, a T got inserted, right? So that will be insertion mutation. All right. So since it is gene mutation, it will be happening only in a single gene. Okay. Now there are further divisions of the base substitution mutation, which can be transition or transversion. Transition mutation we will call if a purine replaces a purine or a pyrimidine replaces a pyrimidine. Now, see, I hope all of you understand that, right? That in DNA there will be two kinds of bases, purines and pyrimidines. So, what are the purines? Quickly tell me what all purines are there. What are the purines that are present in DNA? Absolutely correct. Adenine and guanine, Rajeshwari. Yes, and what are the pyrimidines? Yes, Purbi, Shubhi, Shamalina, Avishka. Correct. Adenine and guanine are the purines. What are the pyrimidines? Thymine and cytosine. Right? Okay. So transition will be a type of mutation, base substitution, where a purine replaces a purine, a pyrimidine replaces a pyrimidine. Okay. Yet Nayan, we are talking about DNA only. Okay. So uracil will not be there. Jason, Jaya. Okay. So we are talking about only in DNA. So uracil will not be there. Fine. So transition mutation is a mutation from one purine pyrimidine base pair to other purine pyrimidine base. Okay. So one purine pyrimidine base pair is replaced with another purine pyrimidine base. If you see that, what does that mean? That a purine replaces another purine, a pyrimidine replaces another pyrimidine. Okay. So that is what is called as transition mutation. Transversion will be just the opposite. Okay, so where a purine will be replaced by a pyrimidine or a pyrimidine is replaced by a purine. Okay. So a mutation from a purine pyrimidine base pair to pyrimidine purine base pair. So purine getting replaced by pyrimidine, pyrimidine getting replaced by purine. Okay, so that is called as transversion mutation. You guys can see here in the image that if this is transition, so A is replaced by G. Now A and G both are purines. Okay, P and C both are pyrimidines. Okay, so purine replaces a purine, pyrimidine replaces a pyrimidine. It's a transition mutation. Transversion mutation, purine. So C is replaced with G. Now C is a pyrimidine. It is getting replaced by G, which is a purine. Okay, and G is a purine which is getting replaced by C, which is a pyrimidine. Okay, so purine is replaced by pyrimidine, pyrimidine is replaced by purine. This type of mutation or base substitution is called as transversion mutation. Is this absolutely clear, everyone? Come on, send me some responses, send me some BIOs so that we can move ahead. Transition, transversion, is it clear? Okay, great. Now, this base pair substitutions or base substitution mutations, if it happens in the protein coding genes, then it results into some new type of mutations as well. Those mutations are missense mutation, nonsense mutation, neutral mutation, and silent. Now, what are they? Let's talk. Missense mutation. So here, these four types of mutations that I have written, you'll be able to understand if you understand the pathway through which the information travels. Okay? So the information in DNA is converted into RNA via process of transcription, and then this RNA information in RNA via process of translation is converted into protein, right? So the RNA, they have a group of three nucleotides in them, which we call as codons, correct? Where each codon will code for one amino acid, right? Do you all agree to this? I'll just write it here one more time. So this is DNA. DNA on transcription forms RNA. That RNA undergoes translation and forms the protein. Okay. Now this RNA, especially the mRNA, will have a sequence of three nucleotides in it, and that is called as a codon. And each codon, one codon, will code for one amino acid. Okay, so this codon are like codes, all right, where a meaning of a codon is the amino acid that it codes, right? So we call these codons as sense codons if they code for an amino acid. If the codons do not code for any amino acid, then we call them as the stop codon or nonsense codon. Okay. Now, once you have understood this, it will be easy for you to understand this mutation. Missense mutation. Okay. So sense will refer to the meaning of the codon. Okay. So missense means the sense is changed, right? So if there is a change, if there is a base substitution in the DNA, so that the codon is changed, and because of that, the meaning of the codon is changed. Okay. Example, see here. So the codon in the mRNA was AAA, which was coding for life. Now, because of base substitution mutation that is happening here, okay, the codon changed to GAA, and this GAA now no longer codes for lysine but codes for glutamate. Okay, so this type of mutation which changes the codon to another codon that codes for another another amino acid. This type of mutation is called as a missense mutation. Now, what do we have? Next one. Next one is a nonsense mutation. Nonsense mutation. Now, in nonsense mutation is where it converts into a stop codon, okay, a nonsense codon. All right. So, so if a mutation happens here in the codon, which codes for lysine, now it changed to UAA, which is a stop codon. So that will result into nonsense mutation. Okay, and that will, so a sense codon changed to a nonsense codon is called as nonsense mutation. Is it followed, everybody? Missense and nonsense. Everyone, is it followed? Shamalina, which part? Missense mutation or nonsense mutation? Uh, is missense. Okay, missense mutation, Shamalina. It will be called a missense mutation if if the codon is changed, and because of that, the meaning of the codon is changed. Okay, that means we had a codon that was coding for lysine, but now because of the change, we have a codon that codes for glutamate. Okay, so the meaning of the codon is changed. All right, so that is what is called as a missense mutation. Nonsense mutation, when a coding codon is changed to a stop codon. That will be called as a nonsense mutation. Is it followed, everybody? Okay. Next, we have neutral mutation. Neutral mutation is the mutation where the meaning. So this neutral mutation, it can be a subtype of missense mutation. Okay, where the mutation changes the codon. Because of that, the meaning of the codon is also changed. Okay, so it changes a codon to a different meaning codon. All right, but that different meaning codon now codes for a similar chemical properties amino acid with similar chemical properties. Okay, and that is what is called as a neutral mutation. Okay, you see here lysine and arginine. Okay, they are both basic amino acids. All right. So a mutation. So glycine and arginine, they have both similar chemical properties. So a mutation that changes a codon coding for lysine to a codon coding for arginine will be called as a neutral mutation. Do you all follow? Now, why do we call it a neutral mutation here? Is because this is just the amino acid, right? This amino acid sequence in a polypeptide chain is further undergoes the post-translational modifications which result in the formation of a functional protein. Now, during the post-translational modification, these properties, individual properties of this amino acid, help the polypeptide chain to fold in a three-dimensional space so that the protein can get its function, specific function, right? So if we change the amino acid, but we replace it with a chemically similar amino acid, then it will not cause a major change in the structure of the protein, and therefore it will not cause a major change in the function. Okay, that's why this kind of mutation, where a codon is changed to another codon that codes for chemically similar amino acid, is called as neutral mutation. Is it followed, everyone? Now, next set of mutation that we have is the silent mutation. In silent mutation, there is a change in the DNA. There will be a change in the codon also. So here the codon was AAA, here the codon is AAG. Okay, but in our genetic code, what we have is there are 61 coding codons, but we have only 20 amino acids. Okay, so because of this, what happens? There will be multiple codons coding for one amino acid. Those codons, multiple codons coding for one amino acid, are called as synonymous codons. Okay, so any mutation that changes a codon to a synonymous codon, okay, that is called as silent mutation. Silent because there is change, there is mutation in the DNA, there is mutation in the mRNA, okay, but that mutation will not be shown in the amino acid sequence of the body. AAA also codes for lysine, AAG also codes for lysine. Okay, so this type of mutations are called as silent. Are all these four mutations understood and clear, everybody? Next mutation that we are going to talk about is the frame shift mutation. And this frame shift mutation happens as a consequence of insertion or deletion mutation in the protein coding region. Okay? So if one or more base pairs are added or deleted from the protein coding gene, the reading frame of an mRNA can change downstream to the mutation. So I'll just go back and show you this image. We remember here we talked about insertion and deletion. So when you see there is a deletion, so you see there is a gap created, okay, because one nucleotide is removed. But what is the property of the codons? The codons are present in a continuous manner without any overlap, without any gap, without any comma. So even if there is a nucleotide removed, so for our understanding, we have left a gap, but in the RNA, there won't be any gap. Okay, in the DNA also, there won't be any gap. Instead, all this nucleotide following it will shift one position here. Okay, so C will come here, G will come here, C will come here like that. Okay. Similarly, if there is a new nucleotide added, then all the nucleotides after that position, okay, they will shift. All right. Now, when this shifting is happening, that will change the reading frame. Reading frame means the codon. Okay. The codons will be group of three nucleotides. So when this shifting is happening, the grouping changes, right? As the grouping changes, the meaning of the codons will also, right? So that is called as frame shift mutation, where the reading frame changes. All right. Now, what is very important to understand here compared to the other these four types of mutations that we saw and the frame shift mutation is that in all these four mutations, only one codon is affected, right? Only one codon is affected. Rest all the codons, or rest all the meanings of the codons, they remain same, right? But in frame shift mutation, it not only changes the codon where it is happening, this insertion or deletion is happening, but it also changes all the following codons. Okay, whatever comes after it also gets changed. All right, that is what is frame shift. So the consequence of frame shift mutation, it may generate new stop codons, which will result into premature termination of the translation. It may result into longer than normal protein, right, where the stop codon has got mutated or has got changed to a coding codon. And it may also result in significant alteration of the amino acid sequence of a polypeptide. Okay. So comparatively, if we see, yes, frame shift mutations will have more drastic changes compared to the base substitution mutation. Okay. Avishka, in case of silent mutation, a synonymous codon is coming. Okay, a codon which is coding for the same amino acid. But when we talk about neutral mutation, the meaning is changed. Okay, but now it is coding for a chemically similar amino acid, lysine to arginine, for example. But in case of silent mutation, the amino acid will remain lysine only. Do you follow, Avishka? All right. So that was all about gene mutation, guys. Let's move on to chromosomal mutations. So chromosomal mutation, the size of the DNA lesion is big enough so that it affects the structure and also the number of the chromosomes. The types of chromosomal mutation may be deletion, duplication, inversion, and translocation. I'll explain them with the help of the image here. So deletion. I told you deletion will be there in chromosomal mutation as well as in gene mutation. What is the difference? The size of the DNA. Okay. In case of gene mutation, it will be just deletion of a few or one only nucleotide, whereas in case of chromosomal mutation, it will be a deletion of a big segment. Okay. So if we just divide the chromosome in this segment A, B, C, D, E, F, G, H, okay, one thing you understand, these are segments, okay, these are not the genes, all right, we are just divided it into segments to make you understand the process. So if there is a deletion in this segment, the D got deleted completely, okay, so that is called as a deletion mutation, all right, so this D is completely deleted from the gene. Duplication, where a segment is duplicated. Okay, and when it is duplicated, it can be present just next to the original segment on the same chromosome. It may be present just next to the original segment in the same chromosome, but in a reverse orientation. Okay, or it can be present on the same chromosome, but at some distance. Okay. So if the duplicated segment is present just next to the original position, we call it tandem. Tandem basically means adjacent to each other. So tandem duplication, if it is present next to each other but in a reverse orientation, the duplicated segment is present in a reverse orientation, we call it reverse tandem. And if a terminal segment gets duplicated and then present, then we will call it a terminal duplication. Okay. So likewise, the naming of the different duplication types. Simple meaning the segment of the chromosome is duplicated and okay. Now this duplication, it often results into formation of gene family, okay, where the genes have arrived similar genes with similar sequences coding for similar proteins, not identical but similar proteins, and Those are called as the member of the same gene families, and these gene families they result as because of the next type of chromosomal mutation is inversion. Okay. So inversion, as the name suggests, that a segment of the chromosome will be rotated by 180 degree and reattaches at the same position only. Okay. This inversion can be of two types: paracentric and pericentric. Paracentric inversion we will call when the inverted segment do not include centromere. Okay, the inverted segment does not include centromere. Fine, that is called as paracentric. So you see BCD is rotated by 180 degrees. So here, because of that, the sequence of the segment will change. Here, the original was A B C D E F G H. Now, because of this inversion, it has become A B C D E F G H. All right. Pericentric inversion when the inverted segment includes centromere. Okay, so that will be called as a pericentric inversion. So pericentric inversion, since the position of the centromere is also changed, so the arm ratio. Okay, so you see here it had a one arm bigger, one arm smaller, but because the pericentric inversion changed the position of the centromere, so it changed the arm ratio. Okay, where both the arms became similar length. Okay, this is just an example. Okay, but because the size, the position of the centromere is changed in the pericentric inversion, so it will change the type of the thing. All right. Now, often people get confused between what is paracentric and which is pericentric. Let me tell you. Just remember this IC. IC. IC is there in pericentric inversion and it means includes centromere. Can you people remember this? So just remember IC. IC comes in pericentric inversion, which means includes centromere. Right, everybody? Some responses, guys. Do you all follow so far? Paracentric, pericentric inversion. Okay, thank you, Shubhi, Avishka. All right, moving on. Next, we are going to talk about translocation, which is also a type of chromosomal mutation. Translocation. Okay. Location is position. Trans means change. All right. So there is a change in the position. Okay. So change in the position of what? Change in the position of a chromosomal segment. Right. So translocation involves movement of chromosomal segment from one chromosome to another. Now, see, there is a similar phenomena that happens not as mutation but as a general process, which we call as crossing over. But crossing over involves movement of genetic material between non-sister chromatids of homologous chromosomes. So when we talk about crossing over, it involves homologous chromosomes, right? But when we talk about translocation, it involves non-homologous chromosomes. Okay, and when there is a movement of genetic material between non-homologous chromosomes, that is when we say mutation. All right. So translocation involves non-homologous chromosomes. Okay. Now translocation can be of two types: reciprocal and non-reciprocal. Reciprocal translocation means there is an exchange between the non-homologous chromosomes. So MNO moves to this part, AB from this chromosome moves to the other part. Okay, that is non-homologous reciprocal translocation. Non-reciprocal, where there's a movement in only one direction. Okay, so one segment moves from one chromosome to a non-homologous chromosome. There is no exchange here. That will be called as non-reciprocal translocation. Now moving on to the numerical changes in the chromosome, where the number of the chromosome changes. Okay, that is, they are of two types, euploidy and aneuploidy. Euploidy, where there is addition or deletion of a complete haploid set of chromosomes, right? So you can see here, this is a diploid organism which has each chromosome present in a pair. So if one chromosome from each of this pair is deleted, then the individual will have only one copy of each chromosome that we call as haploid, right? And that is actually be a monoploidy. Mono means one. Okay, so each chromosome will be present in a single copy. That is called as monoploid. So the entire set of chromosome, entire haploid set of chromosome has been deleted. So monoploidy is a euploidy. Similarly, triploid, where each chromosome is present in group of three. Its entire one haploid set is added to diploid, 2n plus n. So it will be triploid. Tetraploid, 2n plus 2n. Okay, where two haploid sets have been added. Because of that, each chromosome is now present in group of 4. Okay, so like that, the ploidy level will be there. Now, these euploids, they have been exploited, they have been used extensively in agriculture. So, all these seedless fruits, starting from your seedless banana, seedless grapes, seedless watermelons, they are all triploids. Okay? Polyploidy has been extensively exploited in so they do not this because of this triploid condition they cannot form fertile seeds. All right, so that's why they are seedless. So that makes this fruit more palatable for us. And because they do not have seeds, so they cannot undergo sexual reproduction. So in those plants, we have to go for asexual methods or vegetative methods of reproduction. Next, let's talk about aneuploids. So aneuploids, there is gain or loss of one or more chromosome, but not the entire set. In fact, never it will be the entire set. Okay. So if we have a diploid cell which is having two chromosomes, a pair of chromosomes, all right. So if one chromosome is deleted from a single pair, okay, that will be the situation will be 2n minus 1. Okay, one chromosome is deleted, all entire segment is present, then it will be called as a monosomy. Now, how do we remember it? Aneuploids, they will be named with a suffix of zomy, and before that, the number will come. Okay, so what number? 2 minus 1. 2 minus 1 is 1. The word for 1 is mono. So it will be monosomy. If one chromosome is present in copy of 3, where all other chromosomes are present in 2 copies, only one homologous chromosome is present in group of 3, then it will be called as, the situation will be 2n plus 1, one extra chromosome present, right? So 2 plus 1 will be 3, right? So word for 3 is tri, that's why it will be called as tri. Similarly, if two copies of one chromosomal pair is present, then 2n plus 2, so 2 plus 2 4, it will be tetrasomy. If both the chromosome of a pair is missing, okay, so 2n minus 2, 2 minus 2 0, word for 0 is null, so null is 0. Okay, so that is how the nomenclature is done in case of aneuploidy. Now here there is something which is double monosomy or double trisomy or tetrasomy. Okay. So in this, what happens is double monosomy. That means two chromosomes are missing, but these two chromosomes are not homologous pair of each other. They are members of different pair. Okay. So one member from two different pairs are missing, and that is called as double monosomy. Okay? So in the same way, if two different pairs have two extra chromosomes, so it will be called as double tetrasomy. Okay. So here you have to understand that 2n minus 1 minus 1 is not same as 2n minus 2. Okay, we cannot do it mathematically. 2n minus 2 will be nullisomy where both the members of a single homologous pair is missing. 2n minus 1 minus 1 will mean that one chromosome from two different pairs are missing. Is it understood, everyone? Aneuploidy, naming and how to calculate the chromosome number. Is it understood, everybody? Why am I asking? Because there have been questions from here. Okay, questions have been asked regarding this chromosome number related to aneuploidy. So if you understand this formulas here, you will be able to answer those questions. Is it clear, everyone? Hmm. Now, how these aneuploids are formed? Thank you, Shubhi Avishka, for responding. Okay, keep responding, guys, so that we can have an interactive session. All right, Rajeshwari. Okay. So how these aneuploids are formed? These aneuploids are formed because of non-disjunction during meiosis. Okay, non-disjunction. Now, junction means joining. Disjunction means separation. So non-disjunction means no separation. Okay, non-disjunction of chromosomes that happens at the in meiosis results into aneuploids. Okay. So because of that, what will happen? The homologous chromosomes will fail to segregate if non-segregation is happening in the first meiotic division. Okay, so the gametes will end up with two copies of the same chromosome. And if it is if the non-disjunction is happening in the second meiotic division, then it will result in the formation of normal gametes as well as the aneuploidic gametes. Okay. Now, moving on to the causes of mutation. Mutations can be spontaneous, mutation can be induced. Okay. So, as I was asking you initially, that we do not come across mutants that often, right? Every day we do not come across mutants. Okay. But does that mean mutation is not happening? No, mutation is happening. The changes are happening. But what happens in our cell? There exists an extensive repair mechanism to take care of this change, to correct this change. Okay, so mutations can be spontaneous, mutation can be induced. Spontaneous mutations are those mutations which occur suddenly at any point of time, which result as a result of natural changes, okay, as a result of natural biochemical processes that are happening in the cell. So those processes, if they cause a change in the DNA, that will be called as a spontaneous mutation. The causes can be replication error or any chemical reaction happening in the cellular environment. And this spontaneous mutations, the rate of it is very slow. The rate of spontaneous mutation rate is very slow. Okay, but this spontaneous mutations are the one which creates variation in the population, and this variation helps in evolution. Induced mutation, they result because of the changes caused by environmental, chemical, or radiation. Okay? So there will be a positive agent that will cause this mutation, and this positive agents are called as mutagens. Okay? The rate of induced mutation will be very high. Okay? So we can come across the same type of mutation in spontaneous mutation as well as in induced. What will be the difference? The rate of induced mutation will be high, whereas spontaneous mutation will be very slow. Now let's check out the causes of spontaneous mutation. Spontaneous mutation may be caused because of replication error, may be caused because of tautomeric shift of bases, or it may be caused because of spontaneous chemical changes like defurination and deamination. Replication error. Now, in the time of replication, we know that the DNA replication happens semi-conservative, right? There will be a template. On the template, the newly synthesized strand will be made by DNA polymerase. Now, if there is a looping out in the template strand, okay, so this portion looped out. Because of that, the the DNA polymerase will not be able to read it because of the looping out. So it will not add any nucleotide here, and instead it will add a nucleotide corresponding to the next nucleotides in the template. Okay, so when this strand separates, so what we will see that there is a deletion in the new strand. Okay, so if there is looping out happens in the template strand, it will result into deletion in the newly synthesized strand. Now, what about this one? If looping out happens in the newly synthesized strand, okay? So if that happens, then DNA polymerase will not realize that there has been a looping out. So instead, it will keep on adding nucleotides corresponding to the template strand, which ultimately will result into insertion in the new strand. Okay? So deletion or insertion of single nucleotide may happen as a result of replication error, and that will be a spontaneous effect, and that's why it is a spontaneous. Next mutation that can cause a spontaneous is because of the tautomeric shift of the bases. Now, can anyone tell me what are tautomers? Yes, quickly. Can you people tell me what are tautomers? Anybody? What are tautomers? No one? Okay. So I'll tell you, tautomers, you must have read it in chemistry, guys. Anyways, so tautomers are basically they are isomers, okay, where the change is in the position of a hydrogen atom. Okay. Yes, they are isomers, correct, Avishka. So they differ from each other because of the positioning of hydrogen atom. All right. Now, the base pairing that happens between two strands of DNA is hydrogen, right? Now, because of the movement of the hydrogen atom that happens automatically, spontaneously as a result of mutation, sorry, as a result of tautomeric shift, which happens spontaneously, okay, that will result in the formation of tautomers. And those tautomers will have different base pairing preference. Okay, so how the preference changes is that a normal thymine, what do we have? That normal thymine will base pair with adenine, and a normal cytosine will base pair with guanine. Okay. And adenine base pairs with thymine, guanine base pairs with cytosine. But when we talk about the rare form of guanine, so rare form of guanine will not base pair with cytosine. Instead, it will base pair with the other pyramidine, that is. Okay. Similarly, the rare form of adenine will not base pair with thymine, but it will base pair with cytosine. Okay. Similarly, The rare form of cytosine will not base pair with guanine but base pair with the other purine, that is adenine, and the rare form of thymine will base pair with guanine. Okay? So because of this tautomeric shift that happens naturally, spontaneously because of the movement of hydrogen atom, the base pairing preference changes. Okay? And that will result into transition mutation, transition based substitution. Okay. The other spontaneous mutation that can happen because of the chemical changes are depurination or deamination. Now, deamination means there is a removal of an amino group. Okay. So, if cytosine undergoes deamination, it results into uracil. All right. Now, it is now if in the DNA that happens that a cytosine got changed to uracil, so the the cell can directly detect it, easily detect it. Why? Because uracil should not be present in DNA. Okay, so there is a special enzyme in the cell to recognize this change in the uracil, and automatically it will be repaired. In case this uracil is not repaired, then it will result into a transition mutation where C is replaced with T, G is replaced with E. Okay? But in the DNA, cytosine is not always present in cytosine, but it is also present in its derivative form, which is 5-methyl cytosine. Now, this 5-methyl cytosine, if it undergoes deamination, it results in okay? In the cell, there exists no repair mechanism to correct it because the cell is not able to identify whether this thymine is because of deamination of 5-methyl cytosine or is it because yeah, of its position or it is supposed to be present there on because thymine is a part of DNA, right? So deamination of 5-methyl cytosine results into transition mutation, all right, and these regions of DNA, which has high level of 5-methyl cytosine, are called as mutation hotspots, who have higher chances of mutation as compared to any other region. All right. So that is deamination, which can be a cause of spontaneous mutation. Now moving on to the induced mutations. Induced mutations, they are caused by mutagens, mutation causing agents. This mutation causing agents may be radiation or may be radiations like ionizing radiation, X-ray, cosmic rays, non-ionizing radiation like UV rays. This UV rays, they result into mutation by what? By forming pyrimidine dimers. Okay, it forms pyrimidine dimers that that creates a kink in the DNA because there should not be no bond, there should not be any bonding between the between the nitrogen bases of the same DNA strand. But UV rays, it creates pyrimidine dimers between the adjacent pyrimidine residues of the same DNA strand, and that creates a abnormality in the DNA structure, which creates a mutation. Okay? Now coming to the chemical mutagens, they can be base analogs like 5-bromouracil, which is a base analog of thymine. So 5-bromouracil can be inserted in place of thymine. Now this 5-bromouracil is more frequently present in its rare form and common form. Okay, it interchanges between the common and rare form more faster than the natural tautomeric. Okay, so it will result into transition mutation because tautomeric shifts result into transition. So base analogs will also result into transition. Deamination agent like nitrous acid. So nitrous acid can remove an amino group and again it can result into transition mutation. Hydroxylating agent which will add a hydroxyl group and that also results in transition mutation. Alkylating agent like methane sulfonate adds an alkyl group and this all mutagens can result into transition mutation. Now, another group of chemical mutagens called as intercalating agents like proflavin, acridine orange, ethidium bromide. Ethidium bromide, I'm very sure you people have used when you perform uh, agarose gel electrophoresis. So, what does they do? They get intercalated, okay, intercalated between the nitrogen bases of the same strand. Okay, so this intercalation is just like we have talked about the replication error of looping up. Okay. So if this intercalating agent gets in the template strand, then it will result in the insertion of a new nucleotide, okay, in the new strand. All right, so that will cause insertion. If intercalation intercalating agent gets in the newly synthesized strand, then it will result in the deletion of a nucleotide from the segment. Okay, so intercalating agent can result into single base insertion or deletion. Is it followed, everybody? All the different kinds of mutations, all the causes of mutations, spontaneous as well as induced. Is it followed? Is it followed, everybody? Can we move on? Because here we come to the end of mutation and now we will move on to population genetics. Okay. Population and genetics. Now, what is population genetics? Okay, now, if you ask me in population genetics, one very important part is the Hardy-Weinberg equilibrium, and we will see that in today's class, and that part is extremely important from your exam for the common entrance test for Central University, as well as it is important for your CSIR exam as well if you are preparing for it after. So, population genetics, what is it? It describes the behavior of alleles in a population. Okay, no longer we are talking about alleles or genes in a single individual. We are now looking at the group of individuals, which we call as population. By focusing on the forces that can cause the allele frequency to change over time, and that is called as evolution. Now, branch of genetics that is concerned with the evolutionary process of natural selection, genetic drift, mutation, migration, and non-random mutation. Population genetic approaches can be used to understand the consequences of these processes individually or in changes in allele frequencies in a population that do not result in reproductive isolation are called as microevolution. And when it results, when these frequencies change the causes reproductive evolution, reproductive isolation, and because of that, it results in the formation of new species. That causes macroevolution. In population genetics, we often come across two terms: allelic frequency and genotype. What is allelic frequency? It is the relative frequency of an allele at a particular locus in the population, okay, which will be expressed as a fraction or percent. Genotype frequency, it is the relative frequency of a genotype in the population. So, how do we calculate this? Very simple formula. So, genotype frequency, how do we say? So, basically, it will be the number of individuals with a particular genotype divided by the total population. So, if n is the total population, so if we have a gene that has got two alleles, capital A and lowercase a, where capital A is dominant, lowercase a is recessive, then it can form three different genotypes: homozygous dominant, heterozygous, and homozygous recessive. Okay? So Frequency of homozygous dominant will be number of homozygous dominant individual divided by the total population. Frequency of heterozygotes will be number of heterozygous individual divided by total population. The frequency of homozygous recessive will be number of homozygous recessive individuals divided by total population. So that's how we can calculate the genotype. How to calculate the allele frequency? In the same way, just that instead of number of genotypes, we will have the alleles number, right? So frequency of an allele will be given by number of copies of that particular allele in the population divided by number of copies of all alleles at the locus. Okay. So when we have, like in this situation here, you have to understand that if we have a gene which had only two alleles, right? Capital A, lowercase. We will have only two allele frequency of capital A, frequency of lowercase. So frequency of capital A, the dominant allele will be how much? Total number of dominant allele divided by the total number of alleles in the population, right? So each individual is diploid. So each individual, so one individual will have two alleles of a particular gene, right? So total number of alleles in the population will be equal to 2n, twice the population. Okay. Now number of alleles. Okay. So here you have to understand that whoever is homozygous dominant, they will have two dominant allele. Whoever is heterozygous, they will have one dominant allele, one recessive allele. And whoever is homozygous recessive, they will have two recessive, right? So when we are calculating the number of dominant alleles only, so we have to consider the homozygous dominants each having two dominant alleles and the heterozygotes having one allele. Okay? So it will be given by twice the number of homozygous dominants plus the number of heterozygotes. Okay, divided by 2 multiplied by the total. Frequency of the recessive allele in the same way will be given by twice the number of homozygous recessive plus the number of heterozygotes divided by 2. Is it followed, everybody? Do you all get the idea how to calculate the allele frequency and genotype frequency? Now, we talked about population genetics, where we study the genetic structure of the population that changes with the help of the evolutionary forces. Now, what are these evolutionary forces? Mutation, migration, natural selection, genetic drift, and non-random. These are the evolutionary forces, and if these evolutionary forces are acting on the population, then the genetic structure of the population will change. Okay, so reverse, if we consider, if there is none of this evolutionary changes happening, okay, then the population, the genetic structure of the population should remain constant, right? And that is what is Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium says that allele and genotype frequencies in a population will remain constant in absence of disturbing influences. What are these disturbing influences? They are mutation, migration, natural selection, genetic drift, and non-random. Okay? So, what are the assumptions of a Hardy-Weinberg equilibrium? First, there is no mutation. Okay? That means in the gene, the gene pool is never modified. There is no introduction of any new alleles or genes in the gene. The population is absolutely randomly mating. Okay, that means all the individuals in the population has equal chance of reproduction. Okay, there is no preference, there is no bias in the reproduction. Next, there is no natural selection. Okay, that means all the phenotypes, all the genotypes are neutral. There is no difference in the survival rate. Okay, so there is no natural selection. The population is extremely large. Why? Because when the population is extremely large, then we can cancel out the effect of the genetic drift. Because smaller the population, more likely it is that the allele frequency will fluctuate by chance from one generation to the next. Okay. So to cancel out the effect of genetic drift means the large population is large. And there is no migration, which means there is no gene flow. Okay? So Hardy-Weinberg equilibrium states that for an extremely large population, uh, where all the individuals are randomly mating, the genetic structure of the population will remain constant generation after generation, provided there is no mutation, no migration, and no natural. Okay? So in very simple terms, it means that if we stop any of these evolutionary forces to act on the population, the structure of the population will be constant. Okay? Now, coming to the numericals which are often asked. So, in case of Hardy-Weinberg equilibrium, we say that we consider two terms. T being the frequency of one allele, Q being the frequency of other. Okay? So, if a gene has only two alleles, okay, so if we have a gene that has only two alleles, capital A and lowercase a. So, frequency of capital A plus frequency of lowercase a will be equal to 1, which will be the total number of alleles in the population, right? So if we replace this frequency of capital A with P and replace frequency of lowercase a with Q, so P plus Q will be equal to 1. Okay, that is what is in terms of allele. Now, for genotype frequency, okay, so when two A's are coming together, that is what we call as homozygous dominant, right? So what will be the frequency of homozygous dominant? That will be equal to multiplication of their individual frequency. Probability rule. We have got two combined events occurring together, independent events, right? So if independent events are occurring together, then it will be given by multiplication of their individual probability. Okay. So frequency of A is P, frequency of A is P. So frequency of homozygous dominant will be P square. Okay. Now what do we have? Frequency of heterozygotes. Frequency of heterozygotes will be equal to frequency of capital A multiplied with frequency of lowercase a. Okay. But it can be in the opposite orientation also. Frequency of lowercase a multiplied with frequency of capital. Okay. So it will be PQ, sorry, it will be PQ plus PQ. So it will be 2PQ. So frequency of heterozygote is given by 2. Frequency of homozygous recessive in the similar manner, it will be Q multiplied by Q, that will be equal to Q. Okay. Now, when we add this genotype frequencies, so there are only three genotypes possible in the population. So, addition of their individual genotype frequencies will be equal to. Okay. So, in terms of genotype frequency, it becomes P square plus 2PQ plus Q square equal to 1, where P square is the frequency of homozygous dominant, Q square is the frequency of homozygous recessive, and 2PQ is the frequency of heterozygous. Is it followed, everybody, so far? Because now we are going to go ahead and solve a equation, solve a question related to Hardy-Weinberg equilibrium. Are you people ready for that? Can I have some responses, everybody? All right, Shubhi. Okay. So let's see this question here. Red hair is a recessive trait in a randomly mating population in Hardy-Weinberg equilibrium. Approximately 9% of the individuals are red hair. What is the frequency of heterozygotes? Okay. See, what happens is whenever you, if the question tells you about the individual showing a dominant phenotype, from there you do not get the information because you know that even homozygous dominant as well as heterozygotes will show the dominant phenotype. Okay? But if the question says you that this percentage or this proportion of the population is showing the recessive phenotype, then we know because recessive phenotype is shown by only homozygous, right? So red hair is a recessive trait. Now 9% of the individuals are red haired. What does that mean? This means, so what we have here, we have the frequency of homozygous recessive, which is 9%. If you convert this 9% into decimal, it will be 0.09, right? Frequency of homozygous recessive in terms of Hardy-Weinberg equilibrium, how do we write it as? As Q square, right? Q square is equal to 0.09. So what will be Q? Q will be equal to 0. Now, as not P should be, okay, so you are giving the answer. All right, we'll see. So now we have Q is equal to 0.3. So if Q is equal to 0.3, how much will be P? We know P plus Q is equal to 1. So P will be equal to 1 minus 0.3, that will be equal to 0.7. We have got the value of P, we have got the value of Q. Sorry, we have got the Q, we have got the P, right? So question is frequency of heterozygotes. So frequency of heterozygotes, how do we show it? By 2PQ. Okay, very simple. Substitute the values in 2 multiplied by 0.7 multiplied by 0. That will be equal to 0.42. When you convert it into percentage, it will become okay. The answer will be option. How easy is it, guys? And this can fetch you very easy marks. Easy calculations, just you have to understand the concept. Understood, everyone? Okay. All right, guys. So let's just see some questions. And as usual, I have taken all the questions from your last 7 years of question paper from this topic. So, very first question. UV exposure is a known mutagen. Which of the following mutation occurs due to UV exposure? So, UV exposure results in the formation of pyrimidine dimers, right? So, it results into option C. Answer is option C, formation of thymine dimers. Next question. Turner syndrome is an example of. Turner syndrome results into X0 condition. That means only one X chromosome is present. Okay. So X chromosome should be present in a pair, right? Sex chromosome should be present in a pair. Either it will be XX or XY. But in Turner syndrome, only one sex chromosome is present. Okay. That means the other sex chromosome is absent or missing. Not the entire set, but only one chromosome is absent. So it will be an aneuploidy. I hope there is no confusion here. Next question, gene which increases the frequency of mutation in other gene. Okay, that is called as a mutator gene. Transition type of gene mutation is represented by, so transition, remember, purine replaces purine, pyrimidine replaces pyrimidine. Okay, so if you see all the combinations given here, only this one where A is replaced by G, C is replaced by C. So A replaced by G is purine replaces purine, C replaced by C is pyrimidine replaces pyrimidine. Okay, so that is the only example of. Next, an individual female with Down syndrome is best represented by. Now, what is Down syndrome? Down syndrome is trisomy 21. Okay, trisomy means extra copy of which chromosome? 21 chromosome. Chromosome number 21 is present in an extra copy. That means three copies of chromosome number 21 will be present. Okay, that is what is Down syndrome. So it's a female. So female will have two X chromosomes. All right. What about the autosomes? In a normal cell, there are 44 autosomes, but in Down syndrome, one extra copy of 21 chromosome is present. So it will be 45 chromosomes, right? So a female with Down syndrome will have the condition 45 autosomes plus a pair of X or pair of X. Next question, position effect. Now, even though we did not talk about position effect, so I'll tell you now. So position effect means that when the position of a gene changes, okay, in the genome, that sometimes results in uh, change in its expression. Okay, that is what is called as position. All right. Now, so position effect, which all mutations will result into change in the position. Deletion, no, because the segment is completely deleted. Translocation, yes, when the segment is moved from one chromosome to a non-homologous. And reversion, no. Inversion, okay. If you see this question, position effect, okay, which will be change in the expression of the gene because of change in its position, can result from translocation as well as inversion. Okay, so there is a mistake in the options that I use. Okay, position effect can be caused by translocation as well as. Coming to next question, number of chromosome in a trisomic individual. Okay, trisomy, that means one copy extra. So it will be, so if there are, so trisomy will be 2n plus 1, right? So next question, a trait follows Mendelian inheritance pattern in a population maintaining genetic equilibrium. If 16% of the population showed the recessive trait, then the frequency of heterozygous individuals in the population is. Okay. So 16% shows the recessive trait. So let's have a whiteboard. So what do we have? Q square is given. How much? 16%. Convert it into decimal, it will be 0.16. So that means Q will be equal to 0.4. When we have Q, P will be equal to 1 minus 0.4, that is 0.6, right? And when we have this 0.6, so what was the question? Frequency of heterozygotes in the population. So Q we have as 0.4, P we have as 0.6. So heterozygotes will be 2PQ, that will be equal to 0.48, right? Or 48%, right? Moving on, harmful mutation do not get eliminated from the gene pool because, these harmful mutations they are usually recessive in nature, and this recessive mutation they remain hidden in the heterozygote. Okay? Because most of the harmful mutations they will become recessive and they will be carried by the heterozygous individual because they will be hidden. Okay, so that's why the harmful mutations do not get eliminated. If the frequency, next question, if the frequency of homozygous recessive genotype is 0.26, keeping Hardy-Weinberg law in place, what would be the frequency of dominant? What do we have here again? Recessive genotype. Okay, so we have Q square. Q square given as 0.26. Very easy. Q will be equal to 0.6. So if Q is 0.6, P will be equal to 1 minus 0.6. All right. So guys, this kind of easy square root should be on your fingertips. All right. You should not be needing calculator to do this kind of square. All right. These are very simple, straightforward square. Okay. If you are able to do this calculation are there. All right, everybody. So as Gandhiji said, live as if you were to die tomorrow, learn as if you were to live forever. So always continue this learning process. Keep your aim, keep your focus on the exam, whatever is your aim, keep your focus on it and keep on working towards it. And thank you everyone for joining. One more time, let me show you the information for the BioTecNika app. You can download it from the Google Play Store as well as from iPhone App Store. Please join our BioTecNika Telegram group. The link is given here for more information on all the important things, all the important researches, updates that are going on in the field of biology. Everybody, please give your feedback. Please fill up the feedback form, and you will get the notes of the lecture. Thank you everyone for joining. Bye. Good night.